case studies where you use the computational tools and approaches to address some of the challenges in the discovery. So here I would say, like it's been long been, the need for the accelerator in the cancer therapeutics has been missing, and it's been long needed for long time since, many years. But until recently in 2019, when CFRIT blessed us with a 5-year grant to establish and run the accelerator for the cancer therapeutics area. And I would like to thank, the leadership of the Texas Medical Center, and especially two other people who I see here on the screen, Stan Vatovich and Susan Temelson, who were really key player to make this happen. So we are proud to partner with them, and we are working, and they are key player in this program. So this accelerator program is a really extensive 9-month program where, with the help of the right people, we are confident that we can help the Texas-based cancer therapeutic researchers to achieve their overall success. So when I say right people, so that involves really the pool of talented people, which involves the strategic advisor, the technicians. So every sort of talented people which you could think of as you move along your drug discovery path. So some of the features I would say for the accelerator is it's a really flexible model, and with the blessing from the CPRIT in terms of grant, we don't take any equity. So another advantage we get here is we are in the Texas Medical Center, so we have access to the world-class researchers and clinicians. And most importantly, this program has been modeled on the success of the TMC Acts, which has been successfully running by the Texas Medical Center for many years. So we also, at the same time, understand that not all of the program in the drug discovery might be fit for the accelerator program. So we also have a computational chemistry resources in place where we aim to help and guide the project or program which are in the early hit discovery or the lead optimization stage. So what we have in the computational chemistry resources, well, we have access to the most advanced hardware in place. And also another advantage we get here is we have access to the open source and the commercially available, drug discovery software packages, which includes, Schrodinger software package, which has been mostly widely used by most of the big pharma companies. So as I said, that at this point, we aim to support early hit discovery and lead optimization programs. And how does it work? So a lot of time, like people, ask me, or when I talk to them, they ask me that what would be the point and where and when they should approach us. Well, I say that, this is the drug discovery pipeline, early drug discovery, the pipeline. And I would say that if you are, in any stage of hit discovery and lead optimization stage, we can definitely help you and we can work together. Let's say, for example, when you are in a hit discovery stage and you have a validated target, and also we have a validated assay in place. So, and then if you approach us and if we work together, we can definitely speed up and accelerate the drug discovery process. We can suggest some of the molecules which you can, fast screen yourself. At the same time, the lead optimization, let's say if you have some sort of optimized hit and you are facing challenges in terms of, solubility, selectivity, tox, so we can help you and work together to, address those challenges as well. So in the next slide, I'll just show you a kind of comparative view that how the traditional workflow works, and how if you involve the competition tool, you can really speed up the process. So the traditional discovery project typically works with either you repurpose the drug molecule, or you purchase a pre-plated set of the diversity compounds, you screen them in your assay system. Again, it depends, how throughput your assay system is. It may take a year or two, and there are many caveats in this this screening, because pre-plated set might be very biased. to some other target. So, people who are in this area would tell you probably if you are getting 1% success rate, you are doing good. At the same time, if you involve the computational, so you let this whole process running and through the computational processes, so you can really fast the process. And instead of screening many thousands of compounds, you could probably screen few hundreds of compounds, and you can 10% success rate is nowadays common, and at the same time you can reduce the timeline. So if you reduce the time, you can not only increase your success rate, but also you can reduce the cost of your program. At the same time, the lead optimization is it's not very straightforward, because when you try to optimize some sort of lead molecule, let's say if you are facing selectivity issue and you sort of address that issue, but now the hard issue comes up.
Yeah.
Then when you address the hard problem, then some tox issue, solubility issue comes up. So what I say, optimize lead is not just removing all the property, but just to keep all the parameter within the threshold. So if all the parameters are within the threshold for any molecule, that is something I would call the optimized lead, and you can move on from this lead optimization to the preclinical stage, taking that molecule. So this was just a high-level overview of the computational resources that we have in place. Also, how computational tools can help you. Then next here, I'll talk few of the case study. I'll talk two case study actually, where in past we use these approaches and address some of the common challenges, or I would say, like most the challenges most of the people face while they work on these projects. So in kinases, we know that most of the kinase, uh, the biggest challenge developing the kinase inhibitor is getting the selectivity. Why? Because the traditional ATP binding pocket is very high. It's highly conserved. And if you look at the molecule, the traditional ATP competitor inhibitors, kinase inhibitors, they all have kind of hinge region where they, like interact with the two of the residues there. And because it's so common, so you would expect that, like the same molecule might hit many other kinases. So you can inhibit basically your kinase, but at the same time, you might be inhibiting other kinases as well, which will be undesirable. So how to address that? Well, let's say here we have protein A and B, and just going back to the problem. So I would say to address that issue, to involve to address and look into the dynamics of the protein, and not just look into the traditional ATP binding pocket, but look for the other pocket which might be unique to that protein. So here, for example, we have protein A and B, and when we run the simulation and when we see the overall RMSD, root mean square deviation, so you don't see much deviation, you don't see much difference. So both protein A and B kind of look the same, not much fluctuation. But at the same time, if you look at some of the loops, so here I'm talking about one of the other activation loop, DLF or DFG activation loop, and yellow is the residue. So basically, in this kind of kinases, there's a DLG, a DLF loop, activation loop. So in the activated state, the activated kinase state, this loop is inward. So basically, phenyl ring is inside the ATP binding loop. So basically, that in that way, it prevents the ATP binding. On the other hand, when it's activated, this loop is on the other side. So phenylalanine is on the other side. And this way, it facilitates the ATP binding. So in some way, if you can find an inhibitor which can possibly interact with this, the aspartic acid of the loop and your target residue, then you can stabilize the out conformation, and in that way, you can actually achieve the inhibition of that kinase. So how do you do that? So let's take into the motion into account. And as you see, when you run the simulation, long simulation, you see that this protein A, this residue is really very flexible. And after some time, it moves closer to the DLG loop and it just stays over there. Whereas in protein B, it doesn't move. So it makes the motion, but it doesn't move over here. So the whole idea, and this graph actually represents the same thing. It goes up and then it stays. Basically, this graph is the distance between the DLG loop and the target residue. So you see that in protein A, this target residues come closer to the loop, and in protein B, it doesn't come closer. So if I would have to choose between protein A and B, I would pick the protein A and try to identify some of the inhibitor which can or ligand which can potentially interact with the DLG loop, as well as they can make the interaction with the residue of interest. So how do we do that? Well, we run the simulation, we capture the dynamics. So let's take, some of the structure where the residue of interest is closer to the loop. And here is why, we wanted to do that, because if you just download one structure from protein database, so that one structure, just one structure represent a snapshot of the protein in given in experimental conditions. So it doesn't represent the whole motion or the dynamic nature of the protein. And these structures, these representative structures are completely different than the protein which you might download from the protein data bank. So you take that these representative structure and then do the virtual screening. So this is just a kind of workflow that how one could do that. This is ensemble-based docking. With many research groups have used this technique, and they have reported more success when they approach and use this approach. So you could use multiple structure, vertical representative structure, and another smart way of doing the process, accelerate the process, is apply the filters before you could do the virtual screening. So you know the pocket now, then you can obviously calculate the volume, polar surface area, how much hydrogen bond acceptor, donor, these kind of parameters or properties which the pocket has it, and apply those parameters to the library before you could submit them for the docking. So this way you can, immediately eliminate some of the unwanted molecule, which might not give you, like meaningful results. So if you apply these filters, you can, from my experience, I can say like, you can remove at least 70 to 80% of the molecule from the big database. So let's say if you start with, 20 million compound, and you will end up, maybe, like kind of 400, 500,000 compounds, so which is manageable, and you can do the virtual screening using those compounds. So the traditional, or I would say that from here on, it's just the conventional way you do the virtual screening, and then once you are done with the virtual screening, the most important part comes. That's prioritizing the molecule for the procurement. Well, for that, again, there's no one path, and there's no straightforward approach. Different people use different approach. Some people use machine learning, some people use the ranking. And it all depends, what information we have already in place for that particular project. So let's say, for example, if we know some of the residues play a very key role in the binding, so we could be biased for those residues. And in this way, we can prioritize some of the hits. If these molecules are already available, commercially available, we can purchase them, or we can synthesize them, and we can experimentally test them. Now, I'm not going in detail of the experimental result, because for this particular target, I don't have the experimental result, but we use the same approach in past, and we have successfully delivered some of the meaningful hits. So this way, actually, you can address, I would say, the selectivity issue among the kinases if you use this approach. In the next case study, I will be talking more about the covalent, designing the covalent, inhibitor, which has been gaining a lot of traction nowadays, especially after the FDA approval of the two covalent drugs. Now, if you look at the surface view of the protein, and this is the ligand, by the way. So this looks like this. It kind of looks. There are many, this one protein and then other effectors, wrapping around it. And then you find a ligand lying in between, the pocket. Now, this is the static structure, but when you look at really in the motion, that how it actually binds, then you see that, all of the things, protein along with the, uh, you know, ligand, water molecule, all are movable objects. They move, but, because of the other forces and interaction with the proteins, ligand stays there. And that's how, it basically, it inhibits the process or accelerates the process, depends on, how it binds to the target. Now, when we talk about the designing of the covalent inhibitors, what would be the strategy, I would say? Well, there could be two strategies. One is if we know that the pocket is druggable and there's a cysteine over there. So you could look for the known inhibitors, in the pocket. When I say known inhibitor, it's not necessarily for the same target, but the family of the same target. Let's say for kinase, so you could scan all of the kinase inhibitor in the same pocket, and then you overlay the structure and see whether, some of the structures are closer to that cysteine, and then basically you attach that warhead to that region and dock it, simulate it, and confirm it before you can experimentally test them. At the same time, you could use the covalent fragments, and then the chances of success, that most of people prefer the covalent fragment. In fact, for the COVID and pro, people are extensively using the covalent fragment because the chances of getting hits is much higher. Although you might be getting like a micromolar affinity or kind of minimal affinity, but you know that those fragment binds, and then once you identify some of the fragment which binds, you can actually grow them to achieve the selectivity and address other issues. So by any means, whatever method you use, the way you have to do it computationally, you have to dock them. And computationally, covalent docking is really computationally expensive and time-taking process as compared to the reversible docking, because it's really two processes involved in that in this covalent interaction. One is the thermodynamics followed by the kinetics. So ligand has to stay for a certain time close to that cysteine so that it can make interaction. So if you compare the traditional reversible docking, it's, I would say, like 10,000 ligand per processor per day. But if you at the same time, if you dock a covalent ligand, it takes 10 minutes, approximately 5 to 10 minutes per molecule. So that would make few hundred, I would say, per day, which is really, really very slow process. So then what you could do, again, you could use a smart approach and kind of mimic the thermodynamic followed by kinetic approach. So what you do, basically, you can mutate the cysteine residue to alanine and do the reversible docking. By this way, you are just, ignoring the cysteine here, and you are just considering the other interaction in place. And then once you're reversible, because it's really fast, reversible docking is computationally fast process. So once you're done with the reversible docking, you select those conformer of the ligand, which are 5 angstrom or 6 angstrom closer to that alanine, because only those ligand where the warhead of the ligand is closer to that, cysteine, that can only make interaction. So if you apply those filter, you can, I would say 90% of the ligands you can get rid of. And then once you have the top 10% of ligand, which are, in the docking showing closer contact with the alanine, you revert it back, you mutate back alanine to cysteine and do the covalent docking. So this way you could be more efficient and fast. Now, the next question is like how you believe on the covalent docking, the next level of the computational validation. So, I would say, again, capture the motion. So let's say for this ligand, again, in the simulation also, you cannot capture the covalent bond formation. But what you can capture is that the retention time, the residence time of your warhead and the cysteine residue. So how long it stays there? If it stays long enough during this short simulation of process, you could be sure, or you could guess that this molecule might involve or might make the covalent interaction. So let's say for here we have 3 ligands in place, ligand 1, 2, 3, and then when we run the simulation, it's short accelerated simulation. And you see here, and the graph basically downside, this basically represents the distance between the warhead and the cysteine residue. So in ligand 1 and 2, you see that this warhead is pretty much stays on in the direction, and the distance is also stays within the limit, I would say 5 to 6 angstrom. But on the other hand, ligand 3 is just goes away, and it just flips on the other side, and it goes to the effector side. So if I have to make the decision about which ligand to procure first or which ligand to synthesize first, I would go with ligand 1 and 2, and I will give preference to even ligand 1 over 2, and then I would not invest time on the ligand 3. And it's really because covalent designing is kind of I would not prefer the kind of commercially, I would not suggest even to go for commercially available, covalent fragments, because they could, sorry, they could be very biased to some of the target. Also, there are some smarter approach where you could use the kind of commercially available kind of amines, and you can, just attach the warhead. Let's say for the acrylamide, you can just use the one-step coupling reaction, and you can make the novel proprietary novel molecule database, which you can claim the IP. So this way, actually, you could use that library, do the screening, and procure some of the hits. So this would be the smart way of prioritizing the hit. So in summary, I'm intentionally just keeping it in a one sentence that, and I'm very cautious that of using this sentence, that appropriate computational approaches can guide and accelerate the drug discovery with high success rate. This appropriate term is very important because every target, the strategy could be completely different because we have to take into account what prior information is available and what kind of target we are picking off. So with this, I would like to mention our team. Tom Luby, he's the director of the TMC, and he's also investigator, principal investigator in the ACT program. Lance Black is the associate director in TMC and innovation. Ashley Francis, she's a program manager in the ACT. Sarah Hain, she's an entrepreneur in Dresden. I see her here as well on my screen. Myself, Stan Betovich, and Susan Samuelson. They have been very helpful, and they are also, we are also working with them for this program. With this, I would say thank you, all of you, and please reach out to us if you have any questions. If you think that we can help you, reach out to us, because collaboration or teamwork start with the conversation. Thank you.
Wonderful. Thank you very much, Amit, for getting us started in this area. What we are going to do, what we typically do, is let both speakers get started. And then if you can, as the speakers are giving their presentation, if a question comes to you, go ahead and throw it up in the chat. And at the end of both speakers, we'll start addressing the chat questions, and then kind of open it up if there are audience questions. At that point, feel free to unmute your mic and engage the speaker. And that way we'll have kind of both speakers able to address some of the questions. I realize some of the questions may be more to one speaker than the other. In the interim, try to remember to mute your mic when you're not, when the speakers are talking, okay? So anyways, we're gonna now turn the presentation and the session over to Raji Denny, who goes by Denny.
Yeah, I go by Denny, yeah.
Yep, and from what I understand, first time meeting you, Denny, and it's a pleasure, but you have a number of decades, I won't say many decades, but a few decades, we'll go with that, experience in computational chemistry as focused on drug discovery. You got your master's, PhD degrees, Indian Institute of Technology in Madras, one of the premier institutes in India. Then moved to the US, did your postdoc at Harvard, transitioned from academics more into the industry as a senior principal scientist at Wyeth. And then Wyeth, if I remember correctly, got bought out by Pfizer. And so you led some of the merger of those research teams, which must have been possibly exciting, possibly traumatic at times, but that would be over a beer perhaps. But while you're at Pfizer at this point, you then rose and became chair of the computational chemistry group at Pfizer. And now you've joined Hotspot Therapeutics, where you're senior vice president and head of their computational sciences. And I know Hotspots just recently closed a Series B now, right?
Yeah.
65 million. So congratulations on that accomplishment. That's a huge Series B. So Hotspot, and with your guidance and focus, is doing great things. And so we look forward to hearing about it. So welcome to Texas, virtually.
Yeah, officially, thanks. Thanks to be invited into Texas. I really appreciate that. Zoom is not new to me. So this is how we've been doing our meeting for the last 3 years in Hotspot. From the show of hands in your reaction tab, like how many of you, can you click the reaction and then like say hi? Can you put a wave? I have a couple of questions before I go into my talk. Okay, Stan, I could see you. Yeah, like, okay, a few of them, that's good. Okay, the question that I did not understand, or like I wanted to understand is like, okay, how many of you are chemists or have done chemistry or drug discovery? A show of hands in your reaction tab. Okay. Yeah, fairly good amount of people. So, okay, when I had this talk, oh, sorry, let me stop sharing. Let me share my correct window. So when I put together this talk, like I didn't know what kind of audience to expect. So I'll be upfront on that. So can you see my screen okay? Okay, so what I did is like I prepared the talk for medchem or comchem audience, but I will try my best because like I see less number of people and more general audience. So I thought like, okay, every slide, like I have rich data. So I will try and explain like what is what. I'll try my best. And so now, let's go there, right? Like, okay, the first one is like, okay, what a paradoxical year we had in 2020. I started off as a scientist, now in the board of Hotspot, and then like I look at numbers and I do business development as well. So what a year has been for biotech. It is a painful year, as you all know, like, okay, for the country, for the entire world. It is a lot of tragedy in terms of disease, like we have unprecedented year. But if you look at the market or in the biotech, there is a very good optimism as well. Like, so such a paradoxical year. If you look at the stock market index for biotech, XBI is basically Spider Biotech ETF fund. It just doubled last year. And then large cap NASDAQ biotech index, again, just jumped up. And if you look at the biotech IPOs that went last year, it's basically like, okay, there were 75 deals roughly worth over $14 billion. Compared to last year, it's like, it was only 66 billion. So unprecedented year. And companies that went IPO and first day stock versus 1 month stock versus 1 year stock, 1 month versus 1 year, it's high. Everywhere you look, like, so if you take the stock market or the biotech industry, like there are lots of diseases that came into light. Like, so I'm just talking about the financial side of things. At the same time, like I'm trying to think as to what it means. Like, what do people feel about it? Like, so it is a very interesting article by Bruce, who's one of my board member in the company. And if you guys are interested, please have a look. Um, it tells like, okay, there is a lot of optimism that drug discovery can cure diseases. That is the fundamental point that I wanted to make here. Like there is plenty of money and there is a lot of job opportunity, a lot of growth potential. Those are all there. And public in general, like if you take up this index, public in general feel very optimistic. Scientists can solve um, problems and get away from this tragic year that we had. So that is very good. With that said, since I just jumbled this slide in when Amit was talking, because like, I didn't know like whether people understand like when I talk about drug discovery, lead generation, and lead optimization. So when you look at any project timeline, um, so first thing is like, okay, you need to identify a good target that you want to work on that is disease relevant, which takes a lot of time. I'm not going to cover that. That takes its own sweet time. It involves a lot of academic group, like doing a lot of CRISPR or like, okay, I intra jivas and lots of uh, studies that needs to get done, understand SNPs, understand the data target. And once you understand, like once you selected a target, that is when all the things that Amit talked about comes into effect. Like we call it like multiple things. In general, it is like, okay, hit generation or lead discovery or hit to lead. Some people call it as hit to lead as well. Normally, it takes about 6 to 9 months. And as a computational chemist or a modeler, what we do is like there are roughly about 42 million compounds. Like when Amit talked about like, okay, case study 1 and case study 2, like virtual screening, he is talking about like, okay, there are 42 million compounds that are available, ready to purchase from vendors. Okay. And what is our objective from a ComChem perspective? In ComChem perspective, the objective is to get a nanomolar, micromolar compound. If you have a micromolar compound, you can do several wonders with it. So that is roughly the criteria that we have. We do virtual screening, we do various other things. Like we want to make sure that target is validated. During this time frame, what we do is like we make sure like all the assays are in place. What are the right assays? Like how we can take the compound to the clinic. Like for it to go to the clinic or drug development stage, there are lots of assays that needs to be there in place. So those are the things that get ready during this lead generation phase. Then this transition is pretty important, like from a funding perspective. So like that means like once you cross this thing, that means like time is ticking. So you generally, like when you go into a lead optimization, that is a topic that I chose to talk here. It is 18 to 24 months. That's very much what you have, 1.5 years to 2 years. Within that time frame is what any company would fund you. And then like, okay, you need to get a compound to the clinic. So you are roughly looking at like, okay, you are either making 2,000 compounds or purchasing it or 20,000 compounds. That's a roughly that much amount is what you can make or you can buy during this phase. During this phase, like, that means like it to lead, like you already selected a good compound or selected 2 or 3 series of compounds. You are trying to optimize like for its potency. In general, like roughly in a nanomolar range, you are trying to optimize it for selectivity. You are making sure it is clearance is there, it is stable, and it is soluble, it is permeable, all those properties, ADME properties, you are trying to make sure it is there in that particular molecule. Then you are throwing 1 or 2 or 3 compounds to the phase 1 study. And phase 1 to phase 3 takes about like 7 years roughly, and different patient population that you're tested on. Okay. And then finally, you end up one as a wonder drug to cure disease and help patients. So that is roughly the timeline. So in that process, like to model a modeler, that's a topic of my discussion. During the lead optimization, what do they really work on? What tools they have at their disposal, and what time what kind of amount of time that they have. So remember, like I put this talk together as for a medicinal chemist or for a computational chemist, but I will try and explain as simple as possible so that like everybody can understand it. Okay. So first, what do modelers work on? This is the data from Pfizer. Pfizer is a company that I worked in for 15 years before joining Hotspot in 2018. So this is the data from Pfizer Worldwide Medicinal Chemistry. WWMC is Worldwide Medicinal Chemistry. So in Pfizer, like, okay, we register all the compounds that why we are making a particular compound. Like we assign why we are making it. So this is about like 320,000 data points. So when we register compounds, like we tell the system why we are making that compound, okay? And I just summarized it, and then like I'm just plotting it here. Potency optimization across all the projects that we worked on. This is a span of 4 years, I think. 55% of the time, this is medicinal chemists. Medicinal chemists are optimizing for potency, okay? We don't know this potency or efficacy that they are trying to get, whether it is for an orthosteric pocket or novel target. What kind of target? I don't know that looking at this. Allosteric pocket or like, okay, phenotypic target. These all have different things. These all mean different things when it comes to computational chemistry, okay? And then like other 45% of the time, either it is selectivity problem that they are trying to solve, or ADME clearance is about 15% of the time that they are trying to solve. Solubility is about 7%, and another 5% for permeability purposes. Okay, so addressing ADME issues implies like, okay, maintaining or improving potency. That means like, okay, they have achieved potency, they want to get selectivity. So that means like they want to maintain this potency and then they want to optimize it. So potency is indirectly involved when they solve these things. Okay, these two are different topic in itself, requires separate time. So I'm just going to pick on potency optimization, and that's what I'm going to talk next. Okay, so so now I've said like, okay, there are multiple different projects, right? So like one is like, it could be orthosteric pocket. This is more like case one that Amit talked about in kinases. Like he was talking about DFG and DLG and other thing. So this is the data that I have on like, okay, 11 internal kinase program. If you don't know, like there are about 52 kinase drugs that are in the market right now. Pfizer, my group was like super efficient. More than 1/4 of them are from Pfizer. So you can probably go and check that. When I put this slide together, like this is 2 years back, like there were about 27 drugs and almost half of them were Pfizer's, I think. So right now, like I didn't check that, but like right now it is about 52, 53 kinase drugs. And I would think like 1/3 or 1/4 should be Pfizer drugs. So we were very efficient in doing kinase drug discovery. So I looked at like internally 11 kinase programs that we worked on for orthosteric pockets. And then like, okay, I looked at like when the project began and when did they first screen the compound, and what is their, how long did it take for them to get 1 nanomolar potency? Okay, they just created the screen, had the screen registered in Pfizer. How long in terms of time it took for them to create the first 1 nanomolar compound or 10 nanomolar compound, okay, to register for that particular project. So when I look at it, about like on an average to get a 10 nanomolar compound, it takes about like 40 to 50 days. So nearly a month and a half or 2 months is what it took from the day the assay is put together for them to get a, nanomolar compound, or 10 nanomolar compound, I would say. Okay, one project took about 100 days, 3 months, 3.5 months, and one project took about 180 days, 6 months to get the 10 nanomolar potency range. So this one is like somewhere around 6 nanomolar. So for us to go after uh, orthosteric pocket, even the worst case, let's say it takes 6 months to get a 10 nanomolar compound, then why does it take for a lead optimization 2 years? That is because, like, okay, it might be potent, but in that same molecule, it needs to have combination of, like, it needs to have clear IP, it needs to have lower clearance. That means like HLM, human liver microsome, it should be stable. It should have so good cellular efficacy. It needs to have lower tox profile. It needs to be lower dose. That is another thing. Tissue distribution, all of those things considered. Okay. So, so if we are not struggling for potency on an orthosteric pocket, okay, then like what we need to have in the modeling tools that we have, okay, tool needs to have less margin of error, because like when a tool that if you have to maintain the potency, and then like you have to optimize for ADME or lowering clearance and other stuff, your tools that you need to have in the computational side of things like need to be solid. It needs to have less margin of error. And now in allosteric pocket are novel targets, like new target that you came up with. When we do potency optimization, are we really struggling? The answer is yes. Unfortunately, I don't have the data. I triaged it, but unfortunately, I don't have the data to show you. Yes, we do struggle for potency. It takes a lot longer to go after a novel pockets or allosteric pockets. In terms of computational chemists or a modeler, need to understand several things, key interactions, like looking at a pocket. What is a key interaction that is going to drive you to get you the potency? That is super important. And they need to have a clear understanding of molecular interactions like hydrophobic effect and desolvation and all the contributions to that pocket, strain energy of the ligand, entropy, enthalpy, and importantly, take into account the almighty water. Water does so many wonderful things in a pocket. Unfortunately, I'm not going to cover that as well in this talk. And then if you are doing phenotypic screening, you don't even know what the target is and you know the pathway. Yes, we do struggle for potency and efficacy. There's an interesting article that was written by one of my colleagues in combination with Genentech and other friends. So I encourage people to look at this. It is a wonderful paper, and it goes over like all the phenotypic project issues. So let's just take up the orthosteric pocket, and then like, okay, let's try and understand this a little bit more, right? So whenever we talk about drug design, what are we trying to achieve in a lead optimization? Okay, the principal goal of the drug design is to lower the human dose and frequency, and for frequency of dose, how often you are dosing for safety and compliance reasons, right? I don't have to say that. But what is the strategy that we need to apply is like, okay, to maximize potency, to minimize the free drug level required for efficacy, and for optimizing PK. So this is, many of you may not have seen this dose equation like the way I've written here. This is called as Wagner's equation. If you are a old-fashioned guy like me, like simply put, like, okay, dose is directly proportional to your potency, and dose is directly proportional to your clearance. Dose is inversely proportional to your bioavailability, and dose is directly proportional to your tau, which is your frequency. Okay, so your potency, if you improve your potency, like if your potency goes down, being like more tightly bound, that means your dose is really small. Okay, we all have heard the statement, dose makes the poison. So lower the dose, like um, better your yeah, better your drug is. Like it gives you a good therapeutic window. And if your clearance goes up, your dose goes up. So you need to keep your clearance down. So and your clearance indirectly affects your bioavailability as well. Okay, so you have to have a check on clearance and your bioavailability. If it goes up, your dose tends to go down, because like if your bioavailability is more, it is inversely proportional, so dose will go down. So that means like you need to have a good soluble compound, permeability, and efficacy, deflux. So all of these things need to be in there. Okay, this is the problem most of the design chemists mistakes that they make. Whenever you make a highly lipophilic compound, it almost always drives the potency. But the problem is like when the potency goes increases, that means like the number goes down, Ki goes down. It increases clearance and it increases tox liability, which is not covered in this equation. Okay, it increases clearance as well. So yeah, you tend to think like, oh, potency is going down, it is good, but your clearance creeps in. So you got to be clever whenever you design compounds. So increasing polarity will preserve potency. It's always a good idea, because like whenever you make selective polarity improvement in your molecule, you tend to have selective compounds, and also like you need to you will keep the preserve a lower clearance. So this clearly dictates like what a modeler should be focusing on. Okay, now what I tell all the young computational chemists is like, when you work with a medicinal chemist, enable compound design with really accurate pharmacological prediction. And that means like log D prediction is good. So like for example, like we no longer run any log D predictions. I don't know whether not. Sorry, let me rephrase that. We no longer calculate any log D, log P, because like our calculations have become so good. We no longer do those things in an experimental setting. So that is a kind of model that we are trying to strive and achieve. Okay, and any binding affinity prediction methods, there is a error margin should be low. That is what we need to have. But what we find is like, for us to do any PK optimization, most of the potency prediction software, you cannot ask the question as to like whether we can maintain that potency. Okay, just to give you why this problem is a little bit more complicated than you think, it's because of this. So let's say imagine two compounds that differ in terms of delta delta G. That is what a computational chemist would calculate. It differs by, if it is 0, like, okay, it is exactly the same or almost same in potency. And your potency of two different compounds is related to your delta delta G of binding using this equation. Okay, so it is related in a logarithmic fashion, or the other way, it is in a exponential fashion. So a delta delta G of 1 means like your potency difference is 5-fold. Okay, and that means like you're talking about like, okay, 0.3 nanomolar to 1.5 nanomolar. It is 5-fold off. But you looked at it in the previous case, your potency is connected to your dose. If all else is the same, one dose will be like this. For this one, the other compound will be like this. Like that is how big a dose should be. So one will be a regular human pill, the other one will be horse pill. One will work, you can take it to the market, you cannot take it to a market. Okay, so keeping your dose require, so this is 1 kcal you're talking about. It is nothing big. So if you imagine 2 and 3, like it will make or break your compound. So keeping your calculation error lower is super important in drug discovery. It is a holy grail. So what tools modelers need to have if you are, I've even narrowed it down for potency on orthosteric pocket. And for us to follow that, like I just put together a simple chart. This is the error log, okay? 1 kcal, 2 kcal, and 3 kcal. This is an accurate calculation, and I'm showing you in fold. When you do docking, that in virtual screening or stuff that Amit talked about, that is up here. And endpoint method is like, okay, slightly sophisticated things like, okay, after you do the docking, you can do calculations like MMGBSAs, like solvation models. And then like, okay, you can do enhanced docking fingerprints, like trying to understand like, okay, whether the fingerprints help you. It is not just simply using the scoring function that the program gives you, but you can do sophisticated things. That will enhance your accuracy. It'll be somewhere between 1 to 2 kcal error. If you do FEP, which is currently the latest technology, you are looking at like 1 kcal error. In any project, the things that we do is virtual screening, that is during the hit ID, that lead discovery stage that Amit talked about, and lead optimization. These are the methods that we end up using, like, okay, library design. We design a lot of libraries, and then like, okay, we'll optimize the functional groups to add polarity so that like the compound will, the clearance is reduced. You do scaffold hopping in case like, okay, your competitor is working on it, or like, okay, they already have an IP on it, like you might want to come and get away from that IP, or you even do an atom swap. So all of them, in terms of timeline, like it takes like FEP takes about a day for one calculation, and virtual screening, you can get it done like in a much faster one compound docking. It's much faster, and then like your endpoint method takes few hours or minutes to hours. And then like the same way, like in your timeline, like if you look at like for scaffold hopping and other stuff, like you need to go to sophisticated calculations. Okay, so there is a growing need in the comchems field to develop new methods that are quicker and more accurate, or as accurate as FEP. Okay, those are the things that will help the medicinal chemist. Okay, so having said that, during lead optimization, what are the kinds of transformation or that medicinal chemist would perform? I'm listing some of the simple ones. They do R group variation, like, okay, let's say like in a molecule, like there is a ring system, and then like they might put some R substitution, like, okay, it could be methyl, chloro, ethyl, and other stuff. They do ring transformation, like, okay, they might do a nitrogen walk all over, or oxygen walk, or sulfur walk, like they do many things like that. They do bond change, like, okay, this is sp2 to sp3. They do much such changes. Ring size modification is common. Ring closure is common to reduce entropy. And chiral transformations, most of the time you end up synthesizing a racemic compound, and then like you end up making a chiral specific compound, and then you try both of them. And you do atom insertions. You want to extend it, go into a specific pocket, so you extend those things. And then you do for these things, like you do atom and bond transformations, like systems like this, to get the clear IP. Okay, how accurately we can predict things changes here. In the latest FEP calculation, these are the two things that are being reported to do very good clear calculation. All of these other things have its own issues. Okay, I can go into ComChem side of things and explain like why it is that, but that is the latest. Okay, so we are doing well in certain things, we are not doing well in certain other areas. Okay, so there are lots of tools that needs to get developed, or people are developing right now, and I will share what is the latest to my knowledge. Okay, so the last one is like time component. So this is the old data. Any computational chemist walking into your organization or your company, what is the time that they have to do real project related modeling? So one of my boss, like in the past, like in 2020, she asked us to write down like every hour that you spend on something in an Excel sheet. And we keep sending it like there are about 20 computational chemists, and we kept sending her the Excel sheet. And like 3 years later, like she just tabulated it, and then like, okay, she just made this pie chart. Pie chart. It is interesting to see, like, okay, 9% of the time we spend on publication or presentation. We put together slides to explain to others like what are the things that we are doing. And 12% of the time we are trying to read some scientific journal, or like, okay, doing some software related, learning some software and methods development. 6% of the time we are in symposia like this one, and then like, okay, 2% in travel, and 6% administrative work, and group meetings another 6%, IT related issues, and vacation and project other 6%. We only have 40% of the time to do project modeling. We basically have less time to do stuff that we are hired to do. That is a reality. And there is a reason why people who move from computational chemistry in academics to industry, they have less time to play around with academic code. Because like there are various reasons. One, like, okay, academic folks, like, okay, they develop good science, but unfortunately, like, okay, it is time consuming to do it on like hundreds or 200 200 molecules. It is a lot harder for a chemist who is working in a computational chemist who is working in a company. Okay. Let me just close with this final slide. Stan, like 5 more minutes.
Yep, 5 more minutes.
So this is a typical project progression chart. This is from Pfizer, obviously. So if you understand this, like, you will. I don't know, like, how many of you realize, like, okay, drug discovery is, in addition to being it is a science, there is a process to it. There is a timeline to it. Okay. So what this chart represents is like, okay, x-axis is obviously time, and y-axis is compound number, and this leading dot here, this is when the compound was made. It goes up, and if you see the chart like this, like that means like that day there were hundreds of compounds that are being deposited in our database. And this axis means like, okay, sorry. So this one means like, okay, when different assays are being run. Okay. So the important point here is like, okay, in 8 months time, your medicinal chemists have made 3,500 compounds. That means 15 compounds a day, roughly. Okay. So for a computational chemist to keep track of that and to be impactful, he or she need to be super efficient and know what compound to make, how to make it, and at what time. Okay. So remember these jumps that happen, right? These are all for a reason. Okay. Let me go to the next slide. So what do medicinal chemists do? During a lead optimization, they've already synthesized a core, and they are just hooking on multiple monomers. In this case, like it is a Suzuki. In this case, this is a Suzuki. This is an amide coupling. Okay. This way, they can quickly make tons and tons of molecules. And if you scan the literature from 96 on till 2017, these are the top 10 pharmaceutical companies that one of my postdoc, who's right now in BMS, did. When he analyzed it, like it keeps growing. This is the kind of molecules your medicinal chemists are making. So that means like in one shot, they can make hundreds of molecules by doing these kind of synthesis. And for us to do these kind of things in a computational setting, it is a lot harder. You can do these kind of PMCs, like, so whenever a computational chemist who's in the project, like he or she needs to understand, like, okay, your medicinal chemist techniques have grown as well. So for you to work with them, like you need to understand, like, what are the things that you need to bring. For example, if you have to run a sophisticated free energy perturbation calculation, that means like cost per compound is like $25 to $40, including the license. If you just consider the processing power fee in Amazon, Amazon cluster, if you have to add the licensing fee for that, it is even more. But these kind of PMCs, 100 compounds, you can get it done in a month and even tested at an average cost per compound of only $40 to $90. Okay, because like your core is already being synthesized, it is in the lead optimization state. Okay, so as a computational chemist, like you need to be wiser, your tools have to be faster, and you need to be able to provide, like, okay, there are hundreds and hundreds of monomers that they can hook on to. Which one is more optimal? And focusing on those things will be much more efficient for a computational chemist to accelerate a project. Okay, so I'll skip a couple of slides and then like, okay, this is again like, okay, free energy calculation, like, okay, this is, you just make one atom transformation and your free energy changes drastically. And when you calculate the free energy, the way it is expressed, like, okay, this is coming from the force field perspective, and this is coming from the ensemble average. That means like your ergodicity. When I look at the literature, I'm just going to skip it fast. Like in general, like, okay, people focus, this is your delta delta G of experiment compared to delta delta G of prediction. If your method is predicting it false positive, that means like you are making the molecule, sorry, false negative. That means like your chemists are making the molecule and then it is being inactive. And these are the things that I am finding a lot. So there is a growing need for us to improve the force field. But what we end up doing is like we are in the academic group, what we end up spending is on false positive. We end up doing a lot more ergodicity problem, that is averaging problem, and we end up making a lot of false positives. So this is my observation in the drug discovery. Okay, so with having said that, like, okay, there are lots of positive. Let me quickly jump to my conclusion. So there are lots of positive positive things that are coming out in the literature recently. Like one of the thing being like technique called as SSFEP. So we need the take home is like we need to have 1 kcal or lower. That is a key cutoff, like for us to be really useful. And there are free energy perturbation, which is the best technique that is out there right now, still needs to be improved. We need a faster technique. And then like, okay, there are techniques that are coming up like SSFEP and other things that are in the academics, like that needs to grow even more. Okay, with having said that, like I'll wrap this up. Thanks, Dan.
Well, thank you very much, Denny. And the good thing is, I know you didn't get through all your slides, but your slides will be available online, and the number of people from this talk will be able to kind of take a look at both your slides, Amit's slide, and if they have questions, your email is there, and you know,
Sure.
they'll certainly get in touch with you, and so your time that you get to spend on research is going to shrink even more. So that's a good thing. So what I'd like to do, in the interest of trying to keep this open and kind of as free-flowing as possible, as opposed to me with my, you know, lovely voice reading questions from the chat, I'm going to invite the people who posted the questions, assuming most of them are still on to actually ask their question and, you know, and address it to one or both of the speakers. Is everyone okay with that? And what I'll ask you to do is unmute, ask your question, and then please go and mute. So Gundeep, you had a question right at the very beginning. Are you still here?
Hi, yeah, I'm here online.
Hi.
Yeah, I had a question about whether you provide the virtual compound libraries for screening, or if you want to do the screening, then what are the resources available at TMC? Do you work on a collaboration basis, or how does it work if we have a target of interest?
I guess this question points to me. So Gandeep, There are a number of commercially available compound database available. It's accessible to anyone. I would name few like Zinc database. You could go and you can just customize it. That what kind of molecules are available commercially. You could download and do the virtual screening. We don't provide any kind of library, but we can point you that if you are interested, that what library you should and where you should get those library available, especially those libraries which are available to everybody.
Okay.
And to your second question, we have a kind of so what kind of assistance we provide. So we can if you are expert or if you know how to do the virtual screening, and if you need some sort of assistance, we can guide you. And then also, if you need us to do the virtual screening, you can reach out to us and we can discuss it more, that, what are your goals, what you have already, and then we can prioritize. And if we figure out that, yes, we can work together, then we could do that as well.
Thank you. And I also just going to point out very quickly, we have a ton of questions in the chat. So everyone who's asking questions, conciseness will be rewarded in the next life, but do your best right now. Catherine, how are you doing? Dr. Shine, Catherine, you still there? If not, I'll ask Catherine's question. So Catherine, was that you? No, okay. So Dr. Shine asked, how do you determine this? How do you determine specificity when you are always docking to the same box? That is, when you get into a competitive molecular environment, that your molecule should always bind only to your target. So does that make sense, the question? It might be for either of you. So it says, how do you determine specificity when you're always docking into the same box? And I think what she's getting at is, you have a site, you're targeting that box for virtual screening or optimization. How do you know there's not another site on the molecule that might be better? I believe that's what she's getting at.
So yeah, go ahead, Danny.
So in general, like, okay, every pocket has a signature. Like, for example, kinase pocket. If you are going after an orthosteric pocket, like the one that I mentioned, orthosteric pocket in a kinase, it is binding to ATP. So that means like it is looking for a certain signature. So looking at a compound, like every medicinal chemist would know, like whether that is going to go into an orthosteric pocket or some other region. So if you're going for an allosteric pocket, that again, like for example, there are lots of protein that get phosphorylated, and then like, okay, it goes and then finds certain regions to bind. So all of the pocket have a certain signature of thing that it's looking for. So that is basically what you're trying to reproduce. So whether all gloves are the same, they are not. Like different gloves have different signatures. So that's a way to think about it. So if your glove has to fit your hand, that means like, okay, you need to have the right width and dimensions for it. So the same thing is what you look for in your compound. But when you do the docking, you are specifically narrowing it down to that particular pocket. So you're asking a different question there for this pocket that I have, or this glove that I have, whether this hand will fit. So your question is very clear. So that's what you are trying to do. So if it fits that, then that's what it reports. It doesn't tell you like, oh, whether it will fit somewhere else. It might. We don't know that.
Yeah, I would just add to that point what Danny mentioned. So I think, Shine, you also wanted to know that if the pocket is orthosteric pocket, how you can achieve the selectivity, if I'm understanding your question correctly. So, it's a very tricky question, and to achieve the selectivity, as I said, like just take example of the kinases, molecule which will have kind of hinge region, it might bind to the ATP binding region, but that would be kind of pan inhibitor. To achieve selectivity, you got to know at some point which will distinct your target property with the other target. I mean, say within the kinase domain. and you have to target that. Let's say some, some kind of other residue which are distinct in one kinase and not present in other kinase. So this is all depend, on the target you're working on and selectivity you wanted to achieve. So you got to get to a point where you can distinct your target to your binding pocket to the other pocket and exploit this. This mean only you can achieve the selectivity, especially in the orchestra.
Thank you. Ashwani, are you there? You had a question? Ashwani. Otherwise, you get to hear me ask it. All right. Ashwani, you there? I may have trouble. All right. So Ashwani's question is the following. It says, we have a, I'm not sure what it is, poor eyes. We have a heteromer dimer. Dimer crystal.
Heteromeric dimer. Yeah.
Heteromeric dimer crystal structure with inhibitor molecule. We're not able to find the electron density, but by mass spectrometry, we know the compound is bound covalently. Do you have any suggestion how to probe identify the compound? Thank you. Your interpretation of that question.
Yeah, I'll take that question if I'm understanding the question correctly. So if you know, you got to know, like the chemical structure of your compound and the target residue which you want to target, whether it's a cysteine, some people try to target lysine as well. So at this point, I would say if your compound contains the warhead and you are seeing in your kind of you said that you don't see the electron density, but mass spec you see the compound bind covalently. So I'm not sure whether you want to explore the other covalent inhibitor or you want to confirm that this molecule binds covalently or not. So with the computational tool right now we have, actually we can't capture the covalent bond formation. As I mentioned, some people try to use the quantum chemistry to capture that phenomenon, but I'm not sure, and I'm not quantum chemist. So, but at the same time, I can say that you can use the simulation to make a model near to the reality where you could wisely guess and confidently guess that this covalent formation could take place.
Okay, so my take on this is twofold. One is like, okay, well, normally whenever you do a covalent drug discovery, your compound has to go and bind to the pocket, then it needs to react. So that means your binding has to go first, then the reactivity. So your reactivity cannot be more than the binding. So if I understand if that is what is happening, that means like, and they couldn't get the compound to crystallize, that means like it is a crystallographic construct that they need to focus on. And then like, okay, if you are seeing that in your mass spec, that means like you need to check like, okay, you can do various things like, okay, labeling certain residues to check whether it is really going into the pocket or near the pocket residues, rather than like sticking to somewhere else in the surface. Like you don't want the reactivity to guide your compound to stick to the protein. You might want to double check.
Yep, I think there's definitely a chance it's just non-specific going on there. So Rulong, are you there? Man, I'm batting 100, aren't I? Or 1,000. Rulong, are you there? Either that or everyone's just very shy. All right, well, I'm not. So this is probably more, a little bit to Amit, but probably for both of you. So Rulong said that on one page of the presentation, we were looking at about 50 nanosecond MD simulations. He thought that was fairly short. And so the question, I think, broader question for both of you is, how do you typically think when you're running an FEP simulation or just an MD simulation, how do you go about choosing the time scale for that?
Yeah, so, you want to start with that?
No, like my take on that is like, okay, what are you trying to solve? Like, are you trying to look for a different pocket altogether? Or you're just using it for a virtual screening? For a virtual screening, like all you're trying to do is like, you cannot screen like 42 million compounds. you are trying to reduce the pool to a manageable set of 400 or 500 compounds. For that purpose, it's like, okay, you don't, if you want to reduce that down that way, like, okay, you don't need to worry about, like, okay, whether it is like, you're just increasing the ensemble of confirmation that you are, so 50 nanosecond is fine. But but if you are trying to identify a pocket that doesn't exist, and like maybe you want to go from a inactive state to a active state, and that active state is what you need to inhibit or activate, for that, yeah, 50 nanosecond for that conformational change is not sufficient. So you need to think about strategies like, okay, steered MD or targeted MD. Those are the kinds of techniques that you would want to use. So it depends on like, what is the problem that you're trying to solve?
Yeah, you explain it precisely. I don't have to say anything.
Okay. All right. So Joanne, I'm counting on you to break my perfect streak and hear your question from you, not me.
Hey, yep, Stan, I'm here.
Hey, Joanne.
Yeah, so my question was originally for Amit, but so you mentioned using the simulation frame grabs for your docking. I was just wondering if you could just kind of briefly explain like how long are you running those simulations, and what are the criteria you're using to choose the frames that you use for the dock? And then after that, if you were able to follow any of the projects to completion so that you knew like how accurate your docking scores were in terms of like the actual in vitro binding score.
Okay, so many questions, but I'll answer them. So for the ensemble docking, usually you pick many representative structures. So when I say representative structure and how you select them, so if you look into my slides, so I run like around 400 nanosecond and then I stopped. The reason why I stopped, because after I would say 250 nanosecond, I could see that target residue actually goes closer to the loop and it just stays over there for next 150 nanosecond. So it's getting convergence over there. So that's why I decided to stop. So I stopped after 400 nanosecond. Then again, while I was selecting the representative structure, I specifically select those because I wanted to capture the inactive state, which is the LG out state. So I wanted to take those structure where this target residue is closer to that loop. So in that time frame, I would say to 150 to 400 nanosecond, I selected few representative structure based on the volume. Also, I based on the RMSD of that particular region. So that was the two criteria, and I selected 5 structures based on that. Did the docking, and then again I prioritized the ligands based on. There are many ways. One is the re-ranking, other ways is the looking into the chemical interaction fingerprint as well as the chemical diversity, because taking account into both of the things is very important, especially if you not know much about the target. If you know much or if there's a prior information available, you could be biased to that information as well. And I would, so if you wanted to follow some of the project, you could actually see some of my publication where I used those technique in past. And also, I would not say that there's a direct correlation with the docking scores. Many people try to correlate, but I would not go for that. But you wanted to see whether you are getting some kind of trend or not. As most of the computational chemists, and you are also medicinal chemist, and so docking is not very reliable, but how smartly you could prioritize the hits, how smartly you can use the other tool in the prioritization, that's make you more successful. And it depends, and it will differ also target to target.
So Denny, kind of extending the question, have you found ensemble screening to be a useful approach, or how useful have you found it for initial hits?
Yeah, so for ensemble docking for allosteric projects, like we were successful. For orthosteric, it's a overkill. That is, that's in short what I have noticed. But to go back to Joanne's question regarding like, okay, docking score correlating with KD, even if they say a sophisticated delta G calculation correlating with KD, I would doubt it. So as Amit said, yeah, like you can know, like, okay, compared to this compound, your benchmark compound, whether it will be potent or not. That we are pretty good at doing right now. And that itself is quite a good taking to get there.
Thank you. So, Raju, you had a question posted?
Yes, sir, I'm here. So I want to ask Dr. Gupta. So uh, when we are going to do a docking based on the protein, so what is the best criteria to make sure that we select the right area for generating the grid box? Is it should be based on the ligand that is already present in a crystal structure, or apart from that, should we depend manually on the residue selection and then give the direction to generate the grid box?
Yeah, well, it all depends on your binding pocket, Raju. So if you want to target the pocket where you already have co-crystal ligand, then binding, you selecting the co-crystallized ligand would be the best strategy. If you don't have the co-crystallized ligand and still you wanted to target the same binding pocket, you could select the some of the residues. The whole idea of selecting the grid box or selecting the pocket is to point the program where you wanted to dock your ligand or do the virtual screening. So
Raju,
the,
let me ask you a question. So the structure that you're starting with, is it a APO or you have a co-crystal structure?
That's what exactly I'm going to say. So, like as you mentioned, if we have orthosteric and allosteric sites, so say if we have already co-crystallized ligand in one of the sites, and if we want to target the other site, so I'm just want to make sure like which one, which have you know criteria should be following that.
So yeah, so that means like you are going into a different pocket where it's basically APO
Yeah.
pocket basically. So that's what you want. So you first of all you need to figure out like what is the key driver for your compound to go and interact there. Like for example, if it is ATP pocket, like we know like okay, it needs to have a hinge interaction. If it is a PDE diestrase, you need to know like it needs to have a Q interaction. If it is a bromodomain, it needs to have an asparagine interaction. So knowing what it is like for your pocket that you're looking at, you need to know which is a key driver residue that you need to interact with. Once you figure that out, the rest is simple. Then that's how you construct your grid.
Okay, thank you. So I have one more question to Dr. Denny. I'll ask the second question also. So you mentioned that when you're doing the drug design transformations, so the more the lipophilicity of a compound, it's easy to reduce the toxicity and permeability, right?
Uh, that's a dangerous strategy, increasing lipophilicity. To yeah, I said the opposite actually.
Okay. So yeah, I might have understood it differently then, because
So
I'm curious to understand like
yeah,
when you're
so
doing.
whenever you increase the lipophilicity, pockets are like, so if you think about like why a compound goes into a pocket, right? Compound doesn't know when it is sitting around in a solvent, hey, hiding in there, it's going to be better. It is driven by lipophilicity, right? Like, okay, it doesn't want to be in a polar environment, like, and it finds it and then like, oh yeah, this is optimal, and it goes and sticks in there. So it is driven by lipophilicity in general. Once it goes in there, like it figures out, oh, it has all the right interaction, so I can gladly sit there and water cannot push me out. So if your lipophilicity is the one that is driving it into the pocket, and you definitely see potency, but it'll be non-specific, it'll be binding to everything. So you don't want that, and those kind of compounds get cleared away pretty fast. So it is not going to be productive for a longer run.
Okay. So the main idea for my question is like when you're doing the optimization of the compounds, do you consider to optimize the compounds relative to the solvent to increase the solubility? Because those are the minor issues that we face, right?
Mhm.
Increasing the solubility.
So basically, think about this, right? Like, okay, when you put a compound in a liquid, like, so it needs to dissolve and then dissipate, and then like, okay, it needs to pass through cell. So that means like there are several things that needs to happen. So if your molecule is flat and then it is stacking up nicely, it is not going to be happy in a solvent. So you need to put in some three-dimensionality to your ligand, and your pocket needs to accept that as well. So you need to design those things and then make sure it is soluble and make sure it is not like, make sure it is permeable as well. Like you don't add too much polarity. So it needs a healthy balance you need to have in a compound.
Okay, great. Thank you.
Okay. And Gandeep, I know you had a question about the TMC resources. Was that answered to your satisfaction earlier or not?
Yeah, thank you very much.
Okay. And then Robert, you had a question.
Yeah, just wanted, since I don't come into this with any computational chemistry background whatsoever, and there are a lot of brand names floating around, is there a difference? I mean, of course, particularly Denny, you got to tell me there's a difference, but like, yeah, well, how big of a difference is there going with one machine learning assistive platform versus another?
Different companies have different flavors of what they are focusing on. In your list of things, like, okay, you mentioned Schrodinger. Schrodinger like spends a lot of money in developing force fields, and they are good in that. So different tools have different. So for a project, whenever for example, like if I'm working on a kinase, and if I'm working on an orthosteric pocket, Schrodinger does a very good job. But if you are working on a different, if you're working on an orthostereo, like, okay, or like my scoring needs to be better, I look for vendor flexibility. So CCG and other companies rank a little higher. Not only recently, like, okay, people are talking about like atom-wise, and then like, okay, AI techniques and other stuff. I would wait to see how it plays out. My personal experience from Pfizer is like we were not successful in Pfizer, but when I moved to Hotspot, that is 3 years back, I didn't have that much financial resources to explore all those things, so I stayed away from it. So maybe they have made a lot of progress, and unfortunately, I don't know right now.
Well, we will have next month 2 talks from AI people in drug discovery, so company and academician. So we'll send you an invite. So
it will be good to know.
So to both of you, and this will be, we'll wrap this up because I'm cognizant of the time. You mentioned things that could be working and should be improved. Are there suggestions to the group of things just that they shouldn't even waste their time on, that basically running X, Y, or Z program or approach is no better than just playing a computer game? So what would come to my mind is predicting toxicity, or predicting perhaps liver toxicity from structure. I haven't seen those. I know companies tout their programs to do that. Are there things that you would just suggest, stay away from unless you just have, nothing better to do with your time?
Yeah, Amit, you can go and I will come back.
Well, I would say, some of the learn from the past, so there are some fingerprint, I would say, if you have any of the, like 2 exposed nitro groups, cyano groups, so if you have them, it's a dangerous fingerprints and pattern, so you should probably get rid of them. So those kind of fingerprint might be easy, you could just inspect. There are certain some models also available. There's a paper, I forgot which year it was published. It was industry collaborative, where they pull out all of the tox data, made the model. So one could use it. So it all depends, with whether you are in the early stage or in the late stage of the hit discovery or the lead optimization. If you are hit discovery, obviously, you are not, it's hit discovery, but I call it, like a removal of the unwanted or junk molecules. So you just get rid of them as many as quickly you can. And when you are in the lead optimization stage, I would rather focus on not just one scaffold. In an industry, people usually pursue 4 or 5 scaffold, because you never know which scaffold might, get you success or which scaffold might fail. I can tell you from my experience, when I was in AZ, we were working on the, that was a ADP synthase target, and we pursued 5 scaffold actually. And the scaffold, which was very potent, that actually later on it failed because of the low availability, and also some other issues were there, especially the hug issue. So, so that got failed. And the one which was least potent, that is now in clinical trial with the Evotech. So you never know. And that molecule that was squaramide, there are some of the I would say the pharmacophore which are very good to have it, like the sulfonamides are there, which increase the the hydrophilicity. The squaramides are also good scaffolds, which actually gives you confidence that these are from the past experience with the pharma academic background. These molecules are again and again scaffolds proven to be good as a for the drug candidate. So again, there's no one answer for that. I would say learn from the past and move on.
Yeah. So my take on that is kind of similar, but like the thing is like conventional wisdom is always being challenged. Like for example, 3 years back, if you had said like, oh, I'm going to make a 700 molecular weight compound, okay, in Pfizer or any company for that matter, like people will look up on you as like, hey, it is violating Lipinski's rule, like because it has to be less than 500 for a good oral drug. And now we are talking about PROTACs, which are in 1,000 molecular weight. And there are several companies which are putting compounds in the clinic. So are we questioning it now? Yes. So the important point is like, okay, whether you have set up the right assay and whether you have looked at similar looking compounds that you have tested. So if you have tested similar looking compounds, then your prediction algorithm, whatever that you developed, will help. If your similar compounds is not there in your data set, don't even bother. Like you go generate the data. There is no other way around for it.
Very good. Well, we've hit the top of the hour, and I know, Denny, you're in Boston, and so a little bit later there. So I want to, on behalf of the Gulf Coast Consortium and the people on the call, thank both you and Amit for great presentations and for taking the time. I definitely enjoyed it. So again, thank both of you, and please continue, stay safe, stay healthy, and be good. All right. So thanks very much.
Thank you,
guys.
everybody.
Bye.
Bye.